Mushroom consumption has increased by over 20% in the last decade, with edible mushrooms becoming a staple of modern diets as a result of more plant-based diets, as well as greater understanding of their health benefits and cooking versatility. Over 80% of people enjoy eating these fungal fruiting bodies, which are generally thought of as plants. However, today we'll explore what we're actually eating and how mushrooms are more related to animals than plants. In order to answer what a mushroom actually is, we must look into what a fungus is, for all mushrooms are fungi, but not all fungi are mushrooms. Fungi are eukaryotic organisms, largely differentiated from animals and plants by a cell wall containing chitin. They are abundant worldwide, although often inconspicuous due to the small size of their structures. However, some types of fungi become noticeable when fruiting, either as mushrooms or moulds. Similarly to animals, fungi lack chloroplasts and are heterotrophs, relying on carbon fixed by other organisms for their metabolism. There are three main metabolic behaviours exhibited by the fungi. Mainly, saprotrophic metabolism involves digestion of dead organic matter, which is why mushrooms can often be found growing on dead wood. Fungi can also be parasitic or symbiotic, such as is the case of arbuscular mycorrhiza, which grows inside the root system of plants to receive up to 20% of the plant-fixed carbon, whilst simultaneously increasing the supply of water and nutrients for the host plant. The life cycle of a fungi culminates following sexual or asexual reproduction, which leads to the production of a fruiting body, and in some species, that fruiting body appears as a mushroom. The fruiting body of the fungi contains spores which are often forcibly ejected in order to procreate. Most fungal cells grow as tubular, elongated and filamentous structures known as hyphae. This architecture is adapted for the growth of fungi on or in solid substrates to enable efficient extraction of nutrients thanks to the high surface area to volume ratio. Hyphae are formed by the emergence of new tips along existing hyphae, known as branching, or by the bifurcation of growing hyphal tips. Throughout the lifetime of the fungi, the hyphae continually undergo growth and branching to develop an interconnected network of hyphae, known as a mycelium. The mycelium grows through the substrate and excretes enzymes to convert it into an absorbable form. The larger the mycelium that is produced, the greater access the organism has to nutrients and water, which will enable the organism to produce more fruiting bodies and consequently spread more spores. Hyphae can be septate, meaning it's divided into compartments which are separated by porous membranes called septa. Hyphae may also be aseptate or coenocytic, whereby there are no compartments and the hyphae is essentially a multinucleated supercell. Many specialised hyphae structures exist across the kingdom, such as in Basidiomycetes lilicinus, a carnivorous fungi which is commercially used to kill nematodes that attack plant roots. Before modern science, fungi were considered to be members of the plant kingdom because of their similarities in lifestyle, such as growth in soil and production of fruiting bodies. However, fungi formed their own distinct kingdom and actually diverged from plants and animals around 1 billion years ago. In fact, fungi colonised the land long before land plants. The fungi kingdom falls within the eukarya domain and comprises one subkingdom and 7 phyla. Mushrooms are produced by members of the subkingdom Dikarya, which comprises the 2 phyla Ascomycetes and Basidiomycetes. These species can reproduce both sexually and asexually. Chytridiomycetes are aerobic fungi which can live in freshwater, marine and soil habitats. They produce zoospores, which are motile asexual spores which use a flagellum to move through aqueous phases. Blastocladiomycetes are saprotrophs and parasites of all eukaryotic groups. Neocalymastigomycetes are anaerobic organisms living in the digestive system of larger herbivorous mammals. Also form zoospores, which are posteriorly uniflagellate or polyflagellate. Microsporidia are unicellular parasites of animals and protists. Glomeromycota are the phylum which forms arbuscular mycorrhizae, a symbiotic fungi with plant root cells invaded by fungal hyphae, where both species benefit from the increased supply of nutrients. Ascomycetes are the largest phylum of the fungi kingdom and include some well-known species such as morels and truffles. They are commonly known as sac fungi because their spores are contained within finger-like sacs known as aci. The aci are embedded within the fruiting body called the acicarp, which is formed by a tightly interwoven hyphae network. The asocarps are typically bowl-shaped and can be epigeous, meaning they are found above ground, such as in the case of the morels, or they can be hypogeous, meaning that they're found below the ground, such as in the case of the truffles. Each asi contains 8 asospores, and after dispersal, the asospores may germinate and form a new haploid mycelium. Basidiomycetes form the majority of consumed mushrooms, and the sexual phase begins similarly to Ascomycetes, as compatible haploid hyphae undergo anastomosis to produce a dikaryotic mycelium. The fruiting body develops, which is known as the basidiocarp, and is most commonly known as a mushroom. The basidiocarp has a layer known as the hymenium, which is where basidia generate haploid basidiospores after chirogamy and meiosis. The Basidiomycetes are commonly known as club fungi, since the basidiospores are produced on club-like stalks called basidia. Each basidium typically bears 4 sexual spores. Spore ejection from the basidiocarp is caused by propulsive force derived by a sudden change in the centre of gravity of the discharged spore. A water droplet accumulates at the hilar appendage of each basidiospore, which is where it's attached to the basidium, and that droplet is named Buller's drop. Upon maturity of a basidiospore, sugars present in the cell wall begin to cause the condensation of water vapour in the air on the adaxial face of the spore. When these two bodies of water coalesce, the release of surface tension and sudden change in centre of mass leads to the sudden discharge of the basidiospore. This forcible ejection of spores ensures the exit from the fruiting body and ensures that the spores travel over long distances for effective propagation. These projectile spores are consequently known as ballistospores, since the initial force of acceleration is more than 10,000 times the force of gravity, and the spore is ejected 0.1 to 0.2 millimeters away, which is a lot of distance on these microscopic scales. Some fungi don't make use of ballistospores and instead employ different methods of spore dispersal. For example, the bird's nest fungi uses the force of falling water drops to liberate its spores. Stinkhorns have lively colours and a putrid odour, which attracts insects to disperse their spores. So to summarise everything, the mushrooms that you and I eat are the fruiting body of the fungi. These structures grow incredibly quickly, double in size every day, and represent a very small part of the fungal life cycle. The life cycle starts with the germination of a fungal spore, which occurs under specific environmental conditions, typically involving high humidity and a favourable temperature. The spores grow into haploid hyphae, only containing half the genetic material required to create the new fungus. The hyphae expand into a network called a mycelium, which extracts nutrients and water from the surrounding substrate using enzymes. When two hyphae of the same species combine through anastomosis, it leads to the formation of a fertile mycelium. The mycelium continues to spread through the substrate, and once it's gained enough nutrients, fruiting bodies will begin to form. The environmental conditions largely trigger fruiting, typically necessitating a high humidity and a drop in temperature. This is one of the reasons why mushrooms are most abundant in autumn. A hyphal knot is the first stage of the mushroom, formed when individual strands of hyphae bundle together. Shortly after, clusters of visible bumps form on the surface of the mycelium, which grow into miniature mushrooms known as primordia. Fruiting body selection takes place, with only the healthiest primordia being selected to develop into mature fruiting bodies. As the mushroom matures, gills become visible on the underside of the mushrooms, which contain the basidia and consequently the fungal spores. Some mushrooms have a veil around the stem, which protects the gills during early growth. Eventually, as the mushroom cap grows, the gills become exposed and begin to release the spores. With the release of haploid spores, the cycle can begin once more. That is, of course, if the mushroom wasn't harvested and put on your plates. I hope you enjoyed this video, and if you did, please don't forget to like and subscribe. Join us again next time for some more curious content.